In previous lessons, we learned that cells are the basic units of life. And to remain alive, cells are going to have to do important chemistry within the membrane boundary. And much of that important chemistry involves extracting energy from food. A set of proteins will begin the process, and then the completion of that process happens in a structure called the mitochondria. Mitochondria provide energy then for the cell in the form of chemical energy, which is necessary to perform the chemistry of life. And that chemistry of life includes not only metabolizing food, but also then building important cell molecules. So in this lesson, we'll take a closer look at what's happening in mitochondria. Mitochondria were discovered in the late 1800s, and so here we see some cells, and in fact all these little squiggles would be mitochondria. So there are many mitochondria in a given cell. Here we see a more modern picture, an electron microscope picture. So this is a cross-section of a mitochondria. In this one, we can kind of see that the mitochondria have two membranes. They have an outer membrane, and then the inner membrane folds into these structures that provide extra surface area in the mitochondria. So important chemistry will be happening on the inner membrane of the mitochondria. And having those folds provides more surface area for that chemistry to occur. Now, we previously learned in a simplified way that what's happening in mitochondria is that food molecules are being broken down to extract energy. That chemistry requires oxygen, and of course waste materials are produced from that chemistry, water and carbon dioxide. And the whole point of that chemistry is to produce high energy molecules called ATP molecules. We also learned that ATP molecules have just the right amount of energy for the chemistry that cells are doing. We said that if we imagine glucose to have $20 worth of energy, ATP molecules have $1 worth of energy. And that's significant because a lot of the proteins that make chemical bonds between molecules, they need smaller quantities of energy than glucose provides. So it's handy to have smaller quantities of energy stored in these ATP molecules. Now, if we imagine this is our mitochondria here, then the energy carried in ATP molecules is spent on the chemistry of life. So here we see outside the mitochondria, this would be in the cytoplasm, for example, we'll call that the chemistry of life going on there. Well, the ATP that's being produced in mitochondria will give up its energy to power the chemistry going on in the cytoplasm. When they give up their energy, ATP molecules become ADP molecules, which must be recharged to ATP again in the mitochondria. So ATP molecules give up their energy to power the chemistry, and then the resulting ADP molecules must come back into the mitochondria to be recharged with energy so that the cycle continues. And now let's figure out, how do the ADP molecules get recharged? Well, that's the metabolism of food. So the metabolism of food molecules provides the energy to recharge the ATP molecules. And this chemistry is performed by proteins and requires oxygen. So there's a whole team of proteins here that will be breaking down food molecules, and that process of extracting energy from the food will charge up some certain molecules that we'll name later. And the chemistry releases carbon dioxide waste, and now these energy molecules are going to be coupled. They're going to transfer their energy to ADP to charge ADP up into ATP so that ATP can go out and power the chemistry of life. So in a sense then, what's happening in the mitochondria is that there's an energy transfer from food to ATP. Now that requires complex chemistry, teams of proteins, and that chemistry requires oxygen. And there are a lot of intermediate steps going on here. In fact, this coupling between these energy molecules and ATP is a very interesting coupling, and we can learn about that in another lesson. But the basic idea is that food molecules, the energy stored in food molecules, is being transferred into the energy stored in ATP molecules. Now let's take a closer look and see how ATP is actually used in the cell. We're going to enlist the help of a cartoon biology book called Exploring the Way Life Works. And here we see how life joins reluctant molecules. This big thing here will be a protein, and so it's going to be a protein that will be joining molecules together. First, an enzyme and an ATP molecule form a dynamic duo, a sort of Batman and Robin of the cell, working together to accomplish life's tasks of moving and building. Here we see how they solve the problem of joining two reluctant molecules together. Now let's take a look at step 1. Molecules A and B can bump into each other indefinitely, but will rarely form a bond. They are reluctant partners. So we want to imagine molecules A and B as floating around in the cytoplasm of the cell. So without a protein, these two molecules might bump into each other in the cytoplasm, but rarely will they form a chemical bond. So we'll call them reluctant partners. Now in step 2, an enzyme takes molecule A into its docking site along with an ATP molecule. Now this is the ATP molecule, the very molecule that we said was generated in mitochondria. Right? There was a transfer of energy from food to ATP molecules. So ATP now is out in the cytoplasm ready to give up its energy. So the protein is going to bind molecule A and bind ATP. Now, ATP stands for adenosine triphosphate. And if you look at this molecule, you'll see there are 3 little orange blocks on it. Tri, meaning 3, phosphate means there's 3 of these phosphate groups on the end of the ATP molecule. Now notice what's going to happen in step 2. With careful positioning, the enzyme transfers one of ATP's phosphates to the reluctant molecule. So here we see one of the little orange blocks, that's a phosphate group, has been transferred to molecule A. And that results in an ADP, adenosine diphosphate. 2 phosphates remain. So in step 4 here, the enzyme then discards the rest of the ATP, which in fact is ADP, because it's got 2 phosphates, which is later rebuilt so that it can provide energy for other reactions. So this ADP is the very ADP that has to go back into the mitochondria to get another phosphate group. That's how it's being recharged. So now let's see what happens in the next step. Next, the enzyme takes molecule B into a nearby docking site. So here we have molecule A. Molecule B binds nearby. And what the protein is going to do then, it's going to transfer ultimately the energy of ATP, now the phosphate linked to molecule A, transfer the energy of that chemical bond to make the new chemical bond between molecule A and molecule B. So again, with careful positioning, the enzyme breaks the phosphate off molecule A, simultaneously transferring the energy to a bond between A and B. So the breaking of the bond between molecule A and phosphate releases energy that then is used to make the new chemical bond between molecule A and B. So there's a transfer of energy from one chemical bond into another chemical bond. Now the 2 reluctant molecules are bonded together and the spent phosphate is discarded. Now the spent phosphate will have to go back into the mitochondria, and it'll be floating around in the mitochondria ready to be attached to an ADP molecule to recharge the ADP to make a new ATP molecule, which then leaves the mitochondria, and the cycle goes round and round. So the mitochondria continually produces ATP molecules so that the chemistry of life can occur. But in order to recharge the ADP, to put that phosphate back on to make ATP, food must be constantly broken down. The energy of food must be extracted by proteins, and this complicated chemistry going on in the mitochondria provides a steady supply of ATP for the chemistry of life.